the four institutions we are collaborating with, which include their students, teachers, researchers, and authorities. We jointly seek to motivate them to continue preparing of their professional formation with a universal vision beyond and physical, ideological, and social frontiers. Especially on this day, we celebrate Engineers Day, and this is an ideal way to celebrate it. As a director of this unit, we want to show you some of one Mexico in our city and the National Polytechnic Institute and our unit. I invite you to join us on this date that we address topics and the areas of neuroscience, the immune systems, and biomedical science in the fields of the medical devices and robotics. It's just a window into the great research possibilities you can discover with us. Our unit offers 5 engineering courses, all accredited for their quality by the Accreditation Council for Engineering Education on the international standards. This makes it easier to establish students and teachers exchange agreements between our institutions. Well, the pandemic we are experiencing has prevented us from seeing each other personality, but the doors of the PB are open for all you. Welcome back. I give the words to our moderator, Professor Francesca. Thank you.
Hello, everyone, and welcome on the last day of this summer school. It's my pleasure to introduce the first speaker for today, which will present the cultural session. Dr. Maria Acero is currently head of the research department in the UPB IPN and a biomedical engineering teacher at Instituto Politécnico Nacional. She developed a master's degree on management of technology at Universidad Iberoamericana. After graduating from the Instituto Politécnico Nacional, where she earned a bachelor in biomedical engineering, she entrepreneur self company about biomedical consulting. Later, she joined a national foundation, gained solid experience in assessing developmental and of technology with business opportunity. Afterward, she participates with a research and development company acquiring skills on intellectual property matters and management. She was part of a research program of Instituto Nacional de Rehabilitación, developing a technological transfer model for business opportunities on the health sector. She's also teaching as becoming for her an opportunity for constructing human factor. So it's a pleasure to give the stage to Maria Acero, please.
Thank you, Francesca. Good morning in Mexico, good afternoon in Italy and England. Within the framework of the Global Summer School 2021, I have the pleasure of presenting you the following video named Mexico, Let the Gwellum Sound and Make the UPB Proud, as a sample of the great diversity of our country, Mexico. Moreover, the video will introduce Mexico City, the capital of the country, and the location of the Instituto Politécnico Nacional, our alma mater. Finally, we will end with a presentation of UPB, our university, in order to share with you our areas of development and the opportunity that our students have here. As a general comment, I can tell you that the Gwellum is a hymn of celebration that sounds at each IPN event and makes the heart of our entire community vibrate. We hope you like it, and you are very welcome to visit and enjoy all the things that Mexico City has to offer you. And thank you so much for your attention, and thanks for all those who make this possible. I will share with you this video.
Welcome to Mexico, a country with a great territorial extensive, and due to its geographical position in the world, it has a rich variety of climates. As a result, this has caused through the millions of years of evolution of natural species. Our country is considered one of the 5 most biologically diverse countries in the world. Mexico has 11,122 kilometers of coastlines. It is possible that you have heard about its magnificent beaches where you can travel the way you like, from a great tourism to a walk of a backpacker. The first settlers of the Mexico territory date from approximately 30,000 years ago. The famous pyramids that are mainly found in the central and southern zone of the national territory were created between 200 AD and until shortly before the arrival of the Spaniards. By the way, it is worth mentioning that on August 13, 2021, Mexico will celebrate 500 years of having been conquered by Spain, which should mean a reminder that we are the fusion of 2 civilizations, which the contribution of the Iberian Peninsula in language, musical instrument, and construction, but in turn pre-Hispanic culture contribute ingredients in the gastronomic meals, natural riches, textiles from clothing, as well as create products of pre-Hispanic culture that has been kept alive and expressed daily through different and capricious forms. That is why the same instrument that exists in a symphonic orchestra here it's created the music of the mariachi. It couldn't have happened anywhere else because, as we are proud to say, there is no country same as Mexico. Mexico has a long cultural history with pre-Hispanic colonial representations and each of its stages. From historical reasons, its capital is Mexico City, the place where the main powers are located. Are you ready for a tour? Mexico City has approximately 8 million inhabitants, but daily receives another 4 million who move from peripheral cities of another states to work. However, the character of its people is festive, and you shouldn't miss the opportunities to celebrate everything with multidisciplinary participations, where people dress up according to the occasion and go out to manifest with lots of color, loud music, louder everywhere, and traditional food. Mexico City has one of the most beautiful avenues in the world, the Paseo de la Reforma, where you can find several roundabouts adorned with imposing monuments such as the famous Ángel de la Independencia, meeting center to celebrate any triumph, whether sports or political, and place from which almost all the demonstrations live. One of its most important treasures in Mexico City is the Catedral Metropolitana, which took 300 years to complete, and therefore contains several architectural styles, from the Baroque to the Neoclassical period, all perfectly in harmony. Mexico City is one of the most populous countries in the world, with all the problems that this implies. But it has magic that catches us irremediably. It is yesterday, today, and tomorrow. It is the ground that trembles or the kindness of its people. Perhaps it's all or nothing of the above. But it is love as a living being, as a family member. This is my city of Mexico, and today I have shown you how my heart is inside. In the following video, we present a tour of the Instituto Politécnico Nacional, which, commemorating 84 years of its foundation, shows us its importance within the education of this country. The Unidad Profesional Interdisciplinaria de Biotecnología was founded in 1987. It is located north of Mexico City. Nowadays, its headmistress is Dr. María Guadalupe Ramírez Sotelo. In first place, there is food engineering, a branch of engineering that applies scientific, technological knowledge to design, optimize, and control of preliminary operations, including the processing, production, handling, storage, conservation, control, packaging, and distribution of food products. Then there is environmental engineering. It allows to know the actions to take in order to get a balance between human activities and environmental conservation. Through a teaching based on knowledge, abilities, and competence, where the students can design, install, measure, and evaluate productive systems with social, environmental, and economic benefits, looking for a better life. It also provides methods and facilities and other purposes pertaining to human health and benefit to prevent, control, or remediate any hazards to the environment using their expertise. Biomedical engineering is the application of engineering principles and design concepts to medicine and biology for healthcare purposes. Also included under the scope of biomedical engineering is the management of current medical equipment in hospitals while adhering to relevant industry standards. This involves making equipment recommendations, procurement, routine testing, and preventive maintenance. What about pharmaceutical engineering? It's a career that looks at solutions of design and production of high-quality medicines problems, as well as recovery or production of drugs with emphasis within process design and scrubbing medicines which are used to human and animals. And finally, we have biotechnology engineering. It's a multidisciplinary career dedicated to produce, control, and transform living organisms, their parts, as much as their products, for goods and services purposes, through biotechnology tools. Besides taking over to extend and design equipment and industrial processes where cells or biologic components can act modified by biotechnological techniques in order to acquire higher level products. In addition to being an interdisciplinary institute, the Unidad Profesional Interdisciplinaria de Biotecnología is a cultural center where different activities are carried out, including dance, painting, music, sports, among others. It has hosted events celebrating Mexican traditions, such as Dia de Muertos.
Hi everyone again. I hope you enjoyed the video and see everything that Mexico City has to offer. As you can see in the video, we are the third largest city in the world, so we have many options for you to enjoy. Unfortunately, given the pandemic, it is not possible to meet, but hopefully very soon you will be able to visit Mexico and go to all these amazing places that we see in the video. Mexico is a great city and has a lot of diversity in cultural activities, and it is the base of National Polytechnical Institute, which is our alma mater. The National Polytechnical Institute has many campuses around the city, but now it has some campus outside the city in different states of the Republic. In this case, we are in the UPIBI campus, and Maria will tell us a little bit more about the campus that is the host today. Thank you, Maria.
Thank you, Doctor. Okay, as we can see in the video, UPIBI is a biotechnology unit. We have around 3,000 students in our community, and we are happy to work there. I think it's a very beautiful place where you have a security harmony around the activities. The students can approach the other possibilities of the IPN, like sports or cultural conditions, or actually we have an important international interchange with the students. And I think it's a very good option for students to study in our unit, because they have the opportunity to learn about biotechnology and the applies for our country, and have the opportunity to have a professional development. Dr. Luis, can you tell us what is the opportunities on research that the students have in UPIBI?
Okay, in UPIBI we have 3 graduate programs: productive biotechnology, nanoscience and nanotechnology, and bioprocess. I'm working on the bioprocess sites, which has 3 different lines of investigation. The line that I'm going to present is efficacy and safety of molecules with a potential pharmacological activity. We are 25 researchers in the unit, but we belong to a huge institution. Like, let me explain you a little bit how this works. Instituto Politécnico Nacional is the main education institution, and we have many campuses. One of them is UPIBI, but in total, in National Polytechnical Institute, we are around 1,000 investigators working in several areas of national interest, like development of drugs, environmental science, and so on. What else I can tell you about the graduate program? We have approximately 70 to 80 graduate students. The graduate program started around 15 years ago and has been established according with the measurement of the National Science and Technology in Mexico. Actually, the master degree in bioprocess has been awarded the international distinction, which is the higher grade that CONACYT gives to any graduate program. So UPIBI is a great option if you want to study in Mexico City, and also you will have the chance to see all these amazing things that we see in the video. Thank you, Maria.
Thank you, Doctor. Also, besides the things that you are tell us, actually, the more of the community that we have in UPIBI is they used to live out of the center of the city. I mean, UPIBI is in the center of Mexico, but our community is from another states. So to study here, it's an opportunity to have a personal development, because they are young that starts to live in another house that there is no the parents' house. So it represents a personal development that that construct a community, a stronger community. Besides that, the city offers really good options to have a good place in here. I mean, we have in the video, we can see there are a lot of beautiful places that we can visit in Mexico. So there are a lot of museums. I think you can know that Mexico is one of the most cities in the world that have the most number of museums. We have a lot of national parks. In the Polytechnic Institute, we have the option to have cultural sessions with the Orquesta Sinfónica, and maybe it's a great opportunity to have maybe a very good tour with gastronomic options. What do you think, Dr. Luis, about the gastronomic options that we have in Mexico?
Okay, as we see with our friends from Juriquilla, they tell us about something that is really important in Mexico cuisine, which is tacos. Tacos are known all over the world, and we can proudly say that the best tacos are prepared in Mexico City. The people from Juriquilla probably agree with us. But also we have many options. Today, in the second cultural session, we will talk about one of the main dishes around the country, but it's more focused on the center of the country. It is called pozole. Pozole is a mixture of corn with pork, and which is prepared with many different things, and it depends where region in the country you are. You can find it in many different colors: white, red, green, even black. So in the cultural session, we will show you a video of how a traditional pozole plate is made from the scratch to show you all the process that this important dish is prepared of. What else we have? Well, there are so many options. Also, Mexico has ranking 1 or 2 of the best Latin American restaurants in the world, and it has also, I believe they have at least one of the restaurants in the top 10 restaurants in all over the world. But also, beside the restaurant, there is a lot of, it is common to see around the city, a lot of street food, which is really good. And you don't be afraid to eat in the street in Mexico City. Well, some places, of course, but you can find the most delicious dishes prepared just outside in the street. As you can see, Mexico offers a lot of opportunities, from the poshest restaurants in the world to the best street cuisine also in the world. Additionally, if some of the colleagues are coming to, for example, from Italy, to give a talk here in our institution or in UNAM, we will be able to see different museums, like the Anthropological Museum, which is one of the highest, which is one of the sorry, it's one of the, just some text. One of the let's change it. Let's talk about the music. Music is really important, and as you can see in the video, we see some fragments of mariachis. Mariachis is the regional music, and is composed of different musical instruments, probably around 6 and 7 persons. We are all dressed up in fancy and shiny dresses, and they sung many of the most known songs in the country. Also, National Polytechnical Institute has his anthem, and also has an, let's say, not official song, which is always played in cultural event. So if you are in Mexico City, we can go to our central culture theater and enjoy of mariachis, or enjoy of the Sinfónica, playing one of the most beautiful themes in all over in Mexico. And it is not to brag, but the Mexican national anthem, it is among the most beautiful anthems in the world. I think we are number 4 in the list. So as you can see, we are a really interesting and diverse country, and as the video said, we have a huge population with all of the problems that imply. But every day we try to do our best and try to live in community. And also we are really welcoming with visitors. We like to receive a lot of people, not only in Mexico City, but in our beautiful beaches. Probably have you heard, I know that people in England loves Cancun. Probably they know Cancun better than Mexico City, because it's one of the main holiday destinations. Every year we receive thousands of millions of visitors. We go to our beaches and they return because of our hospitality and our beautiful beaches and really exotic but delicious food. So this was a brief, well, not a brief introduction, because we are talking pretty much for 25 minutes, but we can even be talking more time, because Mexico City has too much to offer that it is difficult to concentrate all of this in 20 minutes. But hopefully this brief introduction gives you a glance of what is Mexico City, and definitely invite you to come not just to the academic sessions or academic activities, but also you have the time to enjoy the variety of cultural activities that this country has to offer. Thank you, Maria.
Thank you, Doctor. As Dr. Luis can say, you will be very welcome, and we will be very happy to see you in an academic convention or in a visit objective. We will be happy to show you the city and the Institute Polytechnic, and obviously we will be happy to receive you at UPIBI and show you what are the opportunity to collaborate with us. Thank you for this session. We can go on the program. We will be happy to see your comments about this item. Thank you.
So thank you to Maria and Luis. And so we're gonna move on to the scientific session. So we can start with the first speaker, Professor Arturo Ortega. Just a brief introduction. Professor Ortega holds a degree in chemistry from the National Autonomous University of Mexico, a master in philosophy with, from CINVESTA, Mexico City, and a PhD in neurobiology from the Weizmann Institute of Science, where he started to work in glutamate-mediated signaling in glial cells. After spending 2 years in the Department of Biochemistry in CINVESTA as an assistant professor, he moved to the Department of Genetics and Molecular Biology, where he was promoted to associate professor in 1997, and in 2004 to the rank of professor. In 2013, he moved to the Department of Toxicology, where he established a neurotoxicology lab. Professor Ortega has been visiting professor in the Institute of Neuroscience of the Autonomous University of Barcelona in 2008, and in the Catholic University of Leuven from 2014 to 2015. He served as a department chair in genetics and molecular biology, and he has been working on glutamate receptor and transporters, focusing on the signaling gene expression regulation at the transcriptional and translational levels for more than 30 years. He has published more than 120 research articles and mentored 36 PhD, 75 master students. He is currently a member of the editorial board of 3 major neurochemistry journals and of the book series Advances in Neurobiology. So with pleasure, I will give the stage to Professor Ortega, and today he will present, he will have a presentation titled Acute Exposure to silica nanoparticle disrupts glutamatergic neurotransmission, role of glial cells. Please, Professor Ortega.
Thank you very much. First of all, I want to thank the organizers for this opportunity. let me see if I managed to do this. You already see my screen, my presentation?
Yes, it is fine, yes.
Okay, so there we go.
Great.
So I was thanking the organizers for letting me, giving me the opportunity to share some of the most recent results from my lab. And since we moved to the toxicology department, we decided that we would undergo, besides our classic neurochemical and neurobiology studies in glutamate receptors and transporters, we decided to focus on a very important health problem, which is air pollution. And in this cartoon, I like this cartoon because it resembles Mexico City in terms that the metropole is a valley, and so being in a valley, you have the accumulation of all the pollutants, which are product both from the vehicles and the industry, which although decades ago the government decided to try to move industrial factories out of the city has grown so much that these industrial parks are already now very close to the city, and in fact in the suburbs. So we decided to collaborate with a colleague, Professor Andrea de Vizcaya, and we decided to try to start to look at the effect of nanoparticles in terms of glutamate transmission. In order to do so, we used two model systems, one of silica nanoparticles, and the other one, which is the one I'm going to focus more today, is of urban airborne fine particle matter named PM2.5 that were isolated and concentrated from the northern part of Mexico City during the spring of 2017. And I want to emphasize that this particulate material was collected in spring because these are this season is the time where pollution is worst in Mexico City. The composition of these particles varies from different parts of the city, but I should tell you that the northern part of Mexico City is one of the areas that has much more pollution, and also it varies between cities, and it varies between seasons because, for example, to collect during the summer is not a very good idea because summer is the time where rain is more present in this area, so you collect very few materials, and in fact you don't have that very strong pollution. So how do these particles get into the brain? Well, inhalation of this contaminated air goes through the nose and goes through the olfactory epithelia and olfactory bulb, and by this means it arrives directly to the brain and gets distributed between a lot of brain areas. I just want to, in order to make the point of my presentation, I just want you to remember a little bit about the brain and the brain cells. So we should remember that the brain is a network of 100 billion neurons, and these neurons connect with at least 100 other neurons in an structure that we call synapse. Okay. We also have to mention that we have 50 times more of other cells which are called glial cells. And these cells, which once were known as supportive cells, have now been discovered to play major roles in the regulation of synaptic transmission. So Another thing that is very important is to remember that the brain is such a complex organ that is isolated from the other parts of the body. And this is because the blood vessels are covered with a bunch of cells which we call neurovascular unit, and this is called the blood-brain barrier. Well, astrocytes, which is a type of glial cells besides oligodendrocytes and microglia, these astrocytes make contact with the blood vessels and with the synaptic terminals. And if I just have told you that glial cells outnumber neurons, in this cartoon is an oversimplified scenario, because turns out that a single astrocyte is has been calculated to engrap more than 10,000 synapses. Okay. The other thing that that I want to put in context is that to understand brain physiology is easy if one assumes that it's a balance between excitation, which means transmission of an electric signal, which is transduced to a chemical signal at the synapse, and inhibition. So if the interaction of the signaling molecule, which is called a neurotransmitter, depolarizes this postsynaptic cell, then we have an excitatory transmission and a continuous signaling. Okay, if the neurotransmitter hyperpolarizes this membrane, the signal is lost. So the brain can be considered as a glutamate machine, and the glutamate is the major excitatory neurotransmitter. And this glutamate machine is regulated by GABA, which is the major inhibitory neurotransmitter, and other less common occurring neurotransmitters, which are quite popular, like dopamine, serotonin, acetylcholine. But all of them are really not very important in terms that they are not very abundant. And finally, we have to say that more than 90% of the brain synapses use glutamate as a neurotransmitter. So here's a cartoon of how a biochemist like myself look at a more closer image to the synapses. This would be which on stimulation would lead to the movement of synaptic vesicles that are going to fuse to the active sites in the presynaptic terminal, and fusing these vesicles would lead to the neurotransmitter release. This neurotransmitter, which is glutamate, is going to act only in the postsynaptic membrane, activating its membrane receptors. And since this is an excitatory signal, glutamate receptors activation would lead to a depolarization of this postsynaptic neuron. Okay, and in order to elicit that, glutamate activates two types of receptors, ones that are ligand-gated ion channels, which upon binding of the neurotransmitter molecule will open sodium and calcium permeable ion channel, leading to an increase in the membrane potential and the depolarization that I just told you. There's another class of receptors which are called metabotropic, that are coupled to GTP binding proteins, but they also, via second messengers, are going to lead to a depolarization. But the faster transmission is responsible. The ligand-gated ion channels are these ionotropic receptors are responsible for this rapid depolarization. So once the glutamate is in the synaptic cleft, it begins to diffuse out of this structure, and the glial cells, which I should emphasize, are more abundant than neurons, and in fact engrap all these synapses, because we are seeing this in 2D, but if you would see this in 3D, this is really embedded in the glial cells, are responsible to remove this transmitter in a very controlled manner. Why is so? Because if you have deficits in the removal of this neurotransmitter is going to continue to depolarize this cell, and this cell will not be able to handle this ionic imbalance, leading to the entry of water and the bursting of this cell and the release of more glutamate to the extracellular medium. Also, you will have a spread of this excitatory transmitter, and it's going to be able to activate other synapses and also extrasynaptic receptors, triggering a phenomena that is called excitotoxicity, which leads to neuronal and oligodendrocyte death, and it's a biochemical signature on most of the neurodegenerative diseases, naming Alzheimer's, Parkinson, all of them. Although the etiology of those diseases are not known, sooner or later you're going to have a dramatic decrease in neuron numbers, which most of them would die through this process of excitotoxicity. On the other hand, if you have more transporters than the ones that are needed, the removal of the neurotransmitter from the synaptic cleft is going to be very fast, and it's going to lead to a decrease in the efficacy of this communication. If you remove this neurotransmitter very fast, you will not have enough time, the transmitter, in order to have this communication. So the molecules involved in this neurotransmitter handling are called glutamate transporters, and from these you have 5 types of transporters named excitatory amino acid transporters 1, 3, 2, 5, and the ones that are expressed in glial cells are E81 and E82, and I should say that, for example, E82 in an adult brain represents 2% of the total protein, which is quite a lot. But the GLAST E81 is more abundant in early stages of development, and also is abundant in the cerebellum, the retina, and the olfactory bulb. So what does these glial cells do that they uptake? Well, once in the air is metabolized to glutamine, glutamine is exported back to the neurons through the SNAT family of transporters. Glutamine, once in the presynaptic neuron, is going to be deaminated in order to generate another time glutamate, and this glutamate is going to be packed in the vesicles, and so there you have a biochemical shuttle known as glutamate-glutamine shuttle that is responsible for the recycling of the transmitter. I should say that if you block this enzyme glutamine synthetase in the astrocytes, glutamatergic neurotransmission shuts down. So this is, for a very good example of a synapse that is not only consistent of the function of a presynaptic and postsynaptic neuron, but also of astrocytes. And these are called tripartite synapses. Okay, so the first thing that we did, once we had the particulate material isolated, as I told you, from the northern part of Mexico City and concentrated and characterized in terms of composition, and we found that it's a major component is silicium. You have also lead, you have also manganese in a complex mixture, and so we decided to expose cultured Bergmann glial cells, cerebellar Bergmann glial cells, which express glass transporter to relevant concentrations of these particles. These concentrations were taken out from the work of a very talented scientist named Garcidueñas, which just reported which would be the actual concentration of the particles in the brain. She worked in, if I'm not mistaken, in a population in Los Angeles, but we took these concentrations out of her reports. So we exposed to different concentrations of this particulate material, and what we found is an increase in the ability of these cells to uptake aspartate, which we use as a non-metabolized analog of glutamate. Because if we use glutamate, it's going to be transformed to glutamine and it's going to be exported from the glial cells. So we found an increase at this concentration. And also we found that this is a time-dependent event. We also, being in a toxicology lab, were very interested to see whether these actual PM concentrations would be cytotoxic. So we did MTT assays and we didn't find any substantial cytotoxicity. So once we said, okay, we have an increase in the uptake of the transmitter, meaning that as a response to this insult, the transmitters, the transporters are functioning in an improved manner, suggesting that the efficacy of the glutamate transmission could be diminished because the half-time life of the transmitter in the synaptic cleft would be diminished. At that stage, we had two possibilities. Either the particulate material would act through the receptor, modifying its structure and leading to an increase in its affinity, or it would lead to an augmentation of the number of transporters within the plasma membrane. I don't have time to go through all the details, but in these proteins, it has been shown that although you have a transcription and a translational regulation, in the short term, in the acute exposure, as the one that I've shown you, that it goes from 5 to 13 minutes, what these transporters do is that they are inserted in the plasma membrane. So in order to gain an insight into that possibility, we did Michaelis-Menten studies. And as you can see, in the material, we have an increase in max with no apparent shift in the Km, suggesting that it would be an increase in the number of transporters in the plasma membrane. In order to prove that in a biochemical way, so we decided to do what are called bioattenuated experiments. So you have your cells, you expose to the material, you do the uptake or whatever the treatment that you want to assay. Then you wash the cells and you put biotin and cross-link biotin to the membrane proteins. Then you make a membrane preparation, you solubilize this membrane, you pull down bioattenuated proteins with the streptavidin, and this pull down, you analyze it in a Western blot with anti-GLAST antibodies, anti-transporter antibodies. So what you see in this
Sorry,
image,
Professor,
yes,
a couple of minutes.
Yes,
Yes,
no problem. So what you can see is that we have an increase in the transporters in the membrane. In order to gain insight to what would be the biochemical process that would be triggered through by the PMs, so we decided to use an antioxidant because in other preparations such as lung cells, it has been shown that particulate matter increases the amount of reactive oxygen species. So we used Trolox as an antioxidant, and we were able to block the increase in the transporter. So we decided to go and look to this process in an immunological way. So you can see that the GLAST is augmented in certain areas of the glial cells, and this is sensitive to the antioxidant Trolax. So in conclusion, we can say that exposure of glial cells to particulate material triggers an increase in ROS. This increase in ROS is linked to an increase in aspartate in Berman glial cells due to an augmentation of transporter molecules in the plasma membrane. And the final conclusion is that this acute exposure modifies glutamatergic transmission, making it less efficient. This is why this has been linked to several deficits in higher brain functions such as learning and memory. So finally, I want to acknowledge the people who did the work. This was work from my postdoc at Dr. Edna Suarez Pozos, my technicians Luis Cid and Luisa Hernandez Kelly, and with the collaboration of Professor Andrea de Vizcaya, and the funding was from the government of Mexico through the National Council of Science and Technology. Thank you very much.
Well, thank you very much, Professor. I have a question from the audience.
Yes.
They are asking which diseases are associated with changes in the transport of glutamate by the presence of particles?
Well, most of the neurodegenerative diseases, as I told you, but the one that has been very linked is ALS, is amyotrophic lateral sclerosis, and this is due to a deficit in the GLT1 subtype of transporter in the spinal cord.
Okay, so thank you for the answer. And no other questions from the audience, it seems like.
Okay.
I thank you very much for a very interesting presentation. And so we will move to the next speaker. Thank you.
Thank you. Bye.
Bye-bye. So the next speaker on the stage will be Dr. José Luis Castrellón. A short introduction. Dr. Castrellón received his bachelor degree in pharmaceutical engineering at the Biotechnology Interdisciplinary Professional Unit, which is UPB in Spanish, of the National Polytechnic Institute. Then he pursued a graded career and obtained a doctoral degree in pharmacology, followed by a short postdoctoral research, both at the MRC Center for Drug Safety at the University of Liverpool, under the direction and supervision of Professor Dean Naisbitt, which is a world leader in drug safety. After finishing his studies, he was incorporated as a full-time investigator at Biotechnology Interdisciplinary Professional Unit since 2011 to the present. Collectively, he adds more than 15 years of experience studying drug safety, particularly those cases involving the activation of the immune system. He counts with several publications in Q1 ranking journal in the field of allergy, immunology, and toxicology. Since he's incorporated to UPB, he has been actively collaborating with other investigators in UPB at the National Polytechnic Institute in different fields, including design and synthesis of molecules for the treatment of bone cancer and the study of biocompatibility of several materials used for the development of prostheses. He is receiving funding by CONACYT, the Mexican Agency for Science and Technology, including Basic Science for Young Investigators and Frontier Science Group, modality in which he has been the investigator leader. He had an administrative role, including coordinator at the master program at UPB, where together with the other investigators belonging to the program obtained the international certification, the highest prize awarded by CONACYT to a graduate program in Mexico. So finally, he has graduated up to 15 master for doctoral studies students and in UPB's graduate program, and currently he is directing for master and 7 doctoral students. So it's my pleasure to give the stage to Dr. Castrillon. Please, 15 minutes for you.
Thanks. Can you hear me and can you see the presentation?
Everything is fine, thank you.
Okay, so let's going to start. First of all, I would like to thank to the committee of the Global Summer event for inviting me to participate. In my talk, we are going to talk about drug safety. Approved drugs have a fine balance between efficacy and safety. In this talk, we will focus on aspects of drug safety and how they impact human health. When a drug causes a harm to the individual who is taking the drug, we call it adverse drug reaction. Adverse drug reactions are an important health problem. It is estimated that 197,000 deaths in the European Union are reported every year due to adverse drug reaction, with a cost to health systems of 5.6 billion annually. Individually, each patient can, the cost of adverse drug reaction can reach up to $8,000. So you can see it's a huge problem with serious economical implications. But what is the classification of adverse drug reactions? Adverse drug reactions are divided into 2 main groups. Type A, which are common reactions that can be predicted, and we know that they are going to happen. Unfortunately, type B reactions, they cannot be predicted, and they have an immunology etiology. These, although are not common, they are related with several life-threatening diseases, as you can see in the picture, which showed you a patient with toxic epidermal necrolysis, a disease caused by a drug and the activation of the immune system with a mortality rate higher than 10%. Also, we have drug reaction and eosinophil syndrome, and severe reaction that has a mortality rate of around 5%. All these type B immune reaction, they will be known in this talk as drug hypersensitivity reaction, or DHR for short. Okay, so I will give you a little bit introduction of what are the cellular molecular mechanisms. This is a complex reaction, so I will show you just a very few frames or very few, not a detailed pathway of how these occur, but hopefully this will help you to understand the pathogenesis of these diseases. So what we have is we take drugs every day. We all take drugs. And what happens is that the drug, once enters the body, it distributes through the body and reach different organs. Once the drug is in different organs, in type B or drug hypersensitivity immune reaction, what is believed that happened, and it has been demonstrated during the last 20 years, is that these drugs somehow activate T cells, conventional and unconventional T cells. The mechanism of recognition, it has been studied, but it is complex, and there are many ways in which a drug can interact and activate the immune system. Once the T cells are activated, they secrete several messengers, in this case, microvesicles, which are in charge of cellular communication. Also, they can secrete cytokines, which have an inflammatory and cytotoxic effect throughout the body. In this talk, we will talk about some of the results that we have on cellular communication, and also some of the results that we have on the interaction of drugs with immune T cell receptors. Also, we will show you just a brief introduction of our new project in which we wanted to know the interaction between the drugs and the microbiota, or the commensal microorganisms that are with us. And we wanted to know if this interaction of crosstalk between microbiota and drugs has an effect in this pathway that I just showed you earlier. So firstly, I will give you a really brief description of how we study this. What we work with the National Institute of Health and Nutrition in Mexico City, and we collect samples from patients, scarce patients, which are the more severe form of the disease, exanthema patients, which are the moderate response to drugs, and tolerant patients, which are defined who are defined as patients who has been under treatment, but they haven't any immune reaction. So we isolate the cell by gradient isolation, and by serial dilution and specific culturing, we obtain drug-specific T cells that are used for the functional in vitro analysis. Then, from the peripheral blood mononuclear cell, we can characterize different type of cell using multiparametric flow cytometry. Finally, we collect the serum, and in this case, we isolate microvesicles in which we perform myrinome and proteome analysis. Okay, so as I was telling you before, this type B reaction cannot be predicted. And another problem with this reaction is that there is not a prognosis biomarker or a preventive biomarker that can tell us that a patient is going to suffer this reaction. Unfortunately, they suffer this severe reaction and they go to the emergency room immediately. So we don't know when this starts until they have the severe reaction. So many of the patients in the field have focused in the study of new biomarkers for drug hypersensitivity reaction. That we have is that recently microRNAs are shown in the picture, are single-stranded 22 nucleotide RNAs, are known as non-coding short RNAs. There are novel biomarkers, and they have been widely studied as a predictive or prognostic biomarkers, particularly in cancer. In 2014, Ishihara demonstrated for the first time that miR-18A is upregulated in the blood of tumor epidermal necrolysis patients. If you remember, those are the most severe form of drug-induced reactions. Not only demonstrate the presence of microRNA 18A, but they also show that the overexpression in vitro of this marker induced apoptosis. Although this was a really interesting and novel investigation, some of the aspects were missing. Is like, for example, do the microRNAs are generated by activated T cells? Are these microRNAs are able to shuttle in the blood to reach other organs? So taking this question, we decide to explore these, looking for these answers. So what we did is what we use in vitro model, which consists in an antigen-presenting cell and a drug-specific T cell from patients, and we stimulated the T cells in the presence of several drugs. Then we measured the expression of microRNA inside the cell, and in a different study, we look for microvesicles from drug hypersensitivity patients, and we characterize their content of microRNAs or microRNAs particularly, and proteins. So I will show you the results of these investigations. What we found is that T cells upregulated microRNAs in the presence of drugs. As you can see in the graphs, in our model system of T cells and antigen-presenting cells, we cultured drug-specific T cells to piperacillin, an antibiotic, and what we see is a high expression of several microRNAs. However, when in the vitro assay we didn't add the piperacillin, we see that there is not activation. The same happened with carbamazepine, another drug implicated with severe cutaneous reaction, and we see the same pattern. Okay. So then we take blood and the serum from drug hypersensitivity patients, and we tried to characterize the microvesicles. Microvesicles, they were thought that they were used just for secreting material that the cell don't use. But recently, it has been found that these microvesicles play an important role in cellular communication. So we isolated the microvesicles, and they were characterized by cryo-SEM. And as you can see, they have round shape with a estimated size of 100 or less than 100 nanometers, which is according with what is seen or the size expected for these particles. Not only we did that, we also checked for membranes of activating or defining activated markers of the microvesicles. And what we found, two of the most important is CD81 and CD9, which are membrane protein exclusively expressed in microvesicles. Okay, so once we characterize the microvesicle, we decide to do a proteomic studies using mass spectrometry. And what we analyze hundreds of proteins, and we see a pattern of several proteins that are preferentially present in a scar patient. However, we just focusing one that found it really interesting. As you can see in the figure, histone H2A was exclusively seen in the DRESS patients using level-free quantification intensity, which is a parameter that the mass spectrometer tells us about the abundance of certain proteins. So it seems that it just expresses in DRESS patient. Nonetheless, we have to we confirm it by Western blot using blood from some of the patients, and as you can see, some of the protein, the histone is present, especially in DRESS patient. This is important because patients with DRESS reaction, once they remitted the disease, they tend to, in the following years, they tend to develop autoimmune diseases. So histone H2A is an important marker for autoimmune diseases. Nonetheless, we have to increase the number of patients and to follow up these patients to see if those with high levels of histone have developed any signs of autoimmune disease. Then we did the Mironome assay using a microRNA assay. As you can see in the picture, we have some, in the heat map, we have some microRNAs which are only expressed in severe patients. So once we obtained this data, we wanted to validate that QRT-PCR. And what we found is that microRNA 18, the one that was found in the previous investigation, it is also seen in the microvesicle. This is really important because if this microRNA travels in the microvesicles, it will have the capacity to reach other cells and other organs. Okay, so that's the first part of the talk when we talk about these possible biomarkers and some of the work that has been done in our lab. Then we move to investigate the role of non-conventional T cells. As I showed you in the slide, in the previous slide, drug hypersensitivity reaction is the result of the activation of normal T cells. Normal T cells are known to have alpha beta T cell receptor, which have a huge variability. But there are other types of T cells called non-conventional T cells, as I show you here, which are mucosal invariant T cells, invariant natural killer T cells, and gamma delta T cells. These cells are characterized because they have a restricted number of T cell receptors. So it's a perfect model to understand how drugs bind to these immune cell receptors. Not only they have a restricted T cell repertory, but they have a high frequency in the skin and the liver, organs in which drug, particularly the liver, is important because drug metabolism occurs there. Also, it has been shown that they recognize small molecules. Actually, in 2017, it was demonstrated that MAIT cells can recognize diclofenac. And the involvement in pathogenesis of drug hypersensitivity is just started to be explored. So in this topic, we wanted to analyze the frequency of MAIT cells in blood of scarce patients. What we found is that the frequency is changed, is altered, but what we can see is that the scarce patients, the frequency of these cells is lower compared with the non-severe form and compared with the healthy donors. However, when we move to the skin, we see that it seems that they are more abundant, particularly in the severe cases. Although the MAIT cells are not that frequent in blood, they have a high activated state, given the high expression of CD107 and interferon gamma. So we were working with this, and as we all know, and that's the reason that we have this virtual meeting, COVID hits the world. And in our case, we have to close the lab. We stopped receiving patients because one of the institutes that works with us has to convert all the resources to treat COVID patients in Mexico. So, but as Albert Einstein said, in the middle of difficulty lies opportunity. So, in collaboration with one of my research colleagues and former undergraduate students with me in the same university, we started to use virtual screening for drug safety. This has been done a bit. A few groups have reported this. So we wanted to learn the techniques and to put it in context with our knowledge. So virtual screening is basically to have 100,000 of molecules and assess them computationally for the capacity to fit in terms of energy and geometric to different receptors. This is routinely done for drug discovery. However, his drug and drug safety has been limited, and only a few groups have started to do this. So how virtual screening occurs? As I told you before, we have a database of compounds, chemical compounds, that can be assessed if we have crystal structures of different receptors. For example, in here I show you the NKT receptor with a crystallographic structure. And what we do is we evaluate how these molecules are energetically and geometrically favorable to interact with this. We can do every compound at a time in a personal computer, but when you have to analyze hundreds of thousands compounds, it is necessary to use supercomputers. And in this case, we use Chiquatl computer from Simvestab. So basically what happens is that we load the structures in the crystallographic structure using specialized softwares and automatized process. Then the computer do all the calculation and show us scoring parameters. Scoring parameters are the result of this interaction, and it can be measured by different for example, by binding energy, by the center of mass, by ligand receptor content distance, and interaction with cryptic amino acids. So once we do this, we wanted to, we analyze this using the Zinc database data versus any crystallographic reference. In this case, we use the crystallographic reference of the 2070 patient, and we analyze up to 4,000 chemical compounds obtained for databases. As you can see, we can discriminate those those drugs or compounds that are far from the pocket of union, or also we can associate them of how these compounds resemble the interaction of the crystallographic structure and the energy. When we compare these two parameters, we end up with this population, which at least in theory, they are favorable to interact with the immune receptor. So after this analysis, these drugs can be potentially evaluated in vitro, and it's a process that we are doing at the time.
Sorry, Dr. Castrillon, just 1 minute ago. Thank you.
Okay, just two more slides, I think. So what is how this can be applied? So probably we can select the receptor, we can do in silico analysis, we can get positive hits, and then structural analysis, and found them with drug hypersensitivity report in drug development to obtain in vitro, to select those compounds that can be candidates to do in vitro analysis. I'm gonna skip this part. And finally, I just wanted to talk to you about some of the new projects that I'm really excited to start. And it is, we wanted to understand what is the relationship between the microorganisms, commensal microorganisms, which are affected by many factors, including diet, disease, age, gender, and so on, and obviously by drugs. So what we wanted to do is, if these drugs interact with the microbiome, and if this crosstalk has an effect on the activation of the immune system. Actually, we are proposing this theory called bacterial immune modulation and type V adverse drug reaction, or VIMBA. So many thanks for the time. We want to thank all these colleagues, which I'm just presenting the work, but actually is the work of many collaboration and many students that has been in my lab. And because of these pandemic times, the meeting has to be virtually. So I wanted to thank you, all of you, for your attention. And if you have any question, let me know.
So thank you very much. I guess we are a little bit long with time, but just a comment with you, Dr. Castrillon, but it seems like that we are more and more looking for interaction between different professionals. So it seems that if we would like to understand and to manage all these knowledges, it seems that biologists and in informatics and other engineering really need to cooperate and to work all together. What do you think about this?
No, yeah, definitely. I mean, I'm just showing the results, but I'm not even close to being expert in many of the fields. So I think this investigation definitely the collaboration of several specialists in different areas. So hopefully this summer meeting will help us to further find topics in common that we can collaborate together. So I think it's very important in different projects and important projects to have a spectrum of specialists.
Just 1 minute, we have a couple of quick
thank you.
Yeah, got it. So one is, how much time takes this virtual essay?
Well, actually, because we have to learn it from scratch, together with my friend, Dr. Carlos Gomez. It took us to set everything and to study to learn how to understand program, computational program, probably 6 months to set the techniques and then run the samples, which they take a few weeks of running or even more. And then you end up with hundreds of thousands of data, and then you have to analyze. I will say 8 to 10 months, starting from scratch, because we have never done it before.
It's quite a long time, but not that long, actually. And another question is, which of the factors you present consider to have the greatest effect on the adverse response?
Consider to have the what, sorry? The
greatest effect on the adverse.
Oh, the greatest effect. That's a difficult one. That's a difficult one.
I think so.
I don't know. I don't know, to be honest. It's because that's the reason we cannot detect them. Even when they're in drug development, that is a huge problem because there are so many factors and they cannot be detected during drug development. Only when it goes to the market, we see the reaction. So it's to give a specific way to each variable, it is really complicated. I don't know. I don't know. Everything contributes, I think.
Yeah, sometimes science is not an exact science.
Yeah, no, there are so many things, but as I say in my talk, is every step counts. So
yeah, exactly.
Hopefully in the future, we will be able to better predict these reactions.
Very much for your interesting presentation and nice interaction here at the end. So actually, as I said, we are a little bit long with the time. So we will go for a short break and we will be back, let's say in 10 minutes. Thank you.
Thank you.